All right, great. Well, thank you, Josh, and thank you, Uma and Erfan, and Bart, of course, for setting this up. I mean, I was super excited when I found out about this. It's a great venue to talk about stuff that I've been interested in for quite a while, and I know I've talked with Bart for years about this stuff. So I wanted to share some of the stuff we're doing, touching briefly on the distant past, but more recent stuff. So I'm going to be focused on the motor cortex and motor control. So I think that sometimes I always got a sense that people view I don't know, there's a lot of focus on vision and cognition, but the motor system and motor control is actually an important aspect of our brains and also of our minds. And I want to share some of this. So also, by the way, the title of this talk, it doesn't imply anything that, we're finding the final solution to coding. I was just playing on the Star Trek theme. So this is, of course, work in progress. So I want to talk about space on the cortical sheet, and particularly in the motor cortex. And I'm sure all of you have seen this diagram, the Penfield map that you see it in all intro neuroscience courses. And where you have a somatotopic map of the legs and the trunk more medially than the arm and hand as you move laterally, and then the face and the tongue and so forth. Oh, wait a sec. So, and this is on a gross level accurate. There's definitely different parts of the motor cortex on a gross level, but if you look at it on a more fine-grained scale, it actually is not. It's misleading. And in fact, Penfield in his papers doesn't make this implication that, for example, that there is a region that encodes the thumb and the index finger and the wrist and so forth. Instead, what you have within a certain body part, whether it's the arm, the leg, or the face, there's a highly distributed pattern of activity. And many studies have shown this. Nevertheless, if you look, particularly in the arm-hand area, you do see some degree of segregation, where the more proximal representations of the shoulder and elbow are represented more medially. So this is an implant. This is in monkeys, macaque monkeys. We've implanted 2 arrays along the central sulcus. The medial array is more represents with intercortical microstimulation. We can evoke movements more often of the shoulder and elbow. And as you move laterally to the second array, you see more reddish and orangish. That's because you elicit movements of the wrist and the hands and the digits. And that generally is true. Although it's highly distributed, and there is no individual thumb area or index finger area and so forth. So we know that, this sort of static view of coding of different body parts in the motor cortex, but can we go beyond that? So does space play a role in coding? And what I mean by that is information about the movement that's being conducted, and not just what body part is being used. And I'm going to break up the talk into 2 parts: movement initiation and execution. I'll start with movement initiation. And I'm going to pose the following question, which has been posed by many groups over the years, is how come movement doesn't occur during motor preparation or action observation? So when you prepare to make a movement, we know that cells in motor cortex activate even before you start moving. And if you impose an instructed delay where you have the animal sit there, wait for a go cue, you see activation in the motor cortex. More strikingly, if you just show the animal or a human, for that matter, an action, very often you'll see activation. So this is work many years ago from my former grad student, Dennis, who showed that activity in many cells are very similar when a monkey performs an action with his arm versus observing the same action on a TV screen. And these are examples of two neurons and the PSTHs of these two neurons. And so if that's the case, and a large part of the motor cortex is activated during observation or preparation, how come you don't move? Because after all, the largest proportion of the cortical spinal tract that activates ultimately motor neurons in the spinal cord come from motor cortex. So there are many hypotheses out there, and I'm not going to go into details about these. There's a gating hypothesis that somehow there's a gate that prevents this action from taking place. There's a threshold hypothesis that maybe you need enough activation to surpass a threshold. And then more recently, Matt Kaufman, who's also here at the University of Chicago, proposed this null space hypothesis, suggesting that because this large population of neurons is high dimensional, there may be subspaces or a null space where the neural population can meander and yet not result in actual movement output. But all of these ideas are missing one thing, which I think is important, which is something that's observed on a mesoscopic scale, which is this oscillatory activity and desynchronization in motor cortex. So the prominent oscillation in the motor cortex, at least in macaque monkeys, and also to some degree in humans, is the beta oscillation. So that's an oscillation that occurs somewhere between 15 hertz and 35 hertz. And I'm showing you here now two traces. On the top row is just a raw local field potential trace during an instructed delay task. So 0 is when the instruction cue comes on, and roughly 2,000 milliseconds later, the movement occurs. And then there's a go cue before the movement indicating, okay, we should start moving. But during that preparation period, before he starts moving, you can see these just in the raw signal, you can see these oscillations very prevalent. And if you band pass the signal into the beta band, you can see them more prominently. So that's nothing new there. It's been documented for decades. And then after the go cue, before movement initiates, you see this dramatic attenuation in the amplitude of the oscillation. So that's all great, but that's nothing new. This is another representation in the power spectrum. Spectrogram. You can see again very prominent beta oscillations during the instructed delay period, and then they shut off right before movement onset. And then they come back on after the movement ends, okay? So if you actually look at the timing at which this attenuation occurs, this decrease in amplitude, you can actually, it correlates very well with reaction time. So this is work done by Vasily, a former postdoc. If you now trial average on a single channel, let's say, trial average the LFP for short reaction times versus longer reaction times and very long reaction times. You can see on the top left, the very short reaction times, you see earlier attenuation of the beta power. For longer reaction times in orange, you see a delayed reaction attenuation. And if you align all these trial average data on movement onset, you see they're all very well aligned at a fixed delay preceding the movement initiation. And this result is shown on the figure on the right, again, that it's fixed with respect to movement onset, but it correlates linearly with reaction time. So we have some signature of movement initiation on a single electrode. One more thing to note. The way I think about this beta attenuation as it's really it represents something like a signature of motor cortical excitability. And this is work done in humans where they used transcranial magnetic stimulation over the motor cortex and applied these pulses and measured EMG activity in the muscles of the thumb, I think it was. And what you find here is as you apply TMS at different periods of time, you see this negative correlation between the excitability, that is the EMG that you evoke by TMS, and the amplitude of the beta power. So in other words, when the beta is low, you have stronger activation of the muscle. So that's the way I view beta attenuation, as a signature of excitability. Okay, so that's all good. But almost 20 years ago, we found there's interesting information spatially, spatio-temporally. So we found the local field potentials, the waves, particularly during preparation, the oscillations during preparation do not form this synchronous sort of standing wave, but rather oscillate in such a way that they propagate across the cortical sheet in time. And we found this was true very consistently, and the propagation direction in the motor cortex. So this is a diagram of the frontal lobe in front of the central sulcus. Right in front of the central sulcus, you have the motor cortex, and each of these double-sided arrows represent 4 monkeys. This is the distribution of directions of propagation, the mean direction of propagation across 4 animals. And they all tend to propagate in one of 2 directions, either rostrally or caudally. I also have 3 other monkeys up here near dorsal premotor cortex that show a slightly different pattern, again, bimodal distribution, but more oriented along the medial lateral dimension as opposed to rostro-caudal. So that was something we saw many years ago, but more recently, we wanted to see what happens with the attenuation when the initiation of movement occurs. And so a former postdoc of mine, Karth, was able to extract single trial waveforms of the beta amplitude and show that in fact the attenuation of these signals is not synchronous across the array. So on the top you see the drop in amplitude of the beta oscillation right after the go cue, before movement onset. And then on the right, color-coded as a heat map, you see the timing at which the amplitude drops below a threshold. And you can see electrodes on the top right attenuate earlier, and those that on the bottom left attenuate later. And we could fit those with planes, and from that we can determine a direction of propagation. And so that's on one trial. This is all representing one trial. The bottom represents a second trial, and we see the same thing except they're propagating in the opposite direction. So mirroring what we saw with the actual phase waves of the oscillation during preparation, the attenuation shows this bimodal distribution of propagation, where this is now a circular distribution of propagation directions, attenuation directions, and you can see a bimodal distribution. So that was kind of surprising to us. We were trying to understand exactly what this means. Certainly it occurs very well timed to right before movement initiation, and it signifies that movement is going to begin. But we try to explore it further. To make a long story short, we found that there was actually no information about what the upcoming movement was going to be performed, but rather it was just signifying initiation of movement. So we went further and we wanted to provide causal evidence that this propagating pattern of excitability or attenuation of beta is a necessary component for initiating movement. And to do that, we use electrical stimulation. So what we did here was we applied stimulation around the time where attenuation occurred after the go cue. And we stimulated not just one electrode, but a whole set of electrodes. So if you look here, if you look at the top row of arrays, you can see we stimulated the let's say on the right side, a whole column of electrodes with a pulse synchronously. Then the second at a certain delay after that, we then stimulated the last column and the last column and the second to last column, and then so forth. So if you go from right to left, you can see how this stimulation is now propagating across the array, and we call that, just for schematic reasons, we'll call that the with propagation. That is the stimulating with the natural propagating attenuation pattern we see in the data. And then we can also apply stimulation in the opposite direction, going against the natural attenuation direction, starting with the left column of electrodes and moving to the right. So we applied that pattern of stimulation right around when the natural pattern of attenuation would occur after the go cue. And to make a long story short, what we found was across 3 monkeys, and regardless of what the monkey was doing, we found a significant slowing of reaction time when we applied electrical stimulation in a patterned way against the natural, that is incongruent with the natural propagating direction of attenuation. That's shown in red. And we see a significant increase in reaction time, anywhere from about 10 to 30 milliseconds, which we don't see with the congruent stimulation. When we apply the same stimulation pattern, the same currents, same electrodes, the same frequency, the only thing is the direction in which we're stimulating. So that was kind of exciting to find that, in fact, this pattern really is important for initiating the movement. One final bit of information is that this axis of attenuation direction, we try to map it onto the somatotopic map. And what you're seeing here is from 3 different monkeys. The blue double-sided arrow is the direction, the propagation axis. And we measured that actually only on the top array. I'm not going to show you on the bottom array, but just on the top array. And you can see it's nearly, not exactly, but nearly orthogonal to the direction of somatotopy going from proximal to distal. Okay? The proximal to distal gradient, as you might call it, is the brown arrow. You're going from shoulder, elbow, down to wrist and fingers. So so it's not exactly orthogonal, but it's interesting that it's chosen this axis, not congruent with a somatotopy. And I have an explanation for that, but we can talk about that later. This pattern of attenuation we see not just with reaching movements, but also with a variety of different movements, including tongue movements, when we had monkeys trained to apply, protrude their tongue and apply a force on a force transducer. We see attenuation in the orofacial area of motor cortex. In C, we see an isometric pinch. We see also attenuation. And in D, we actually had the unique opportunity to look at a human as a part of a brain-computer interface study we're doing. And this was a human out of the University of Pittsburgh, Bard, basically Jen Collinger and Rob Gaunt's group. And this subject actually had some residual movement. So he could actually apply wrist extension movements. And so he applied isometric wrist forces on a force transducer. And we also see an attenuation in the human motor cortex. And moreover, these attenuation signals propagate across the arrays. And so this is the distribution of propagation directions. They're not always equally distributed along the two axes, along the two directions. Sometimes they're highly unimodal, but there's still always a second mode in the opposite direction. So for example, the tongue protrusion, you see very dominant direction of propagation in the caudal lateral direction and a very small one in the rostro-medial direction. But nevertheless, it's always bimodal. Okay, finally, we demonstrated that these propagation directions signal movement initiation, but they don't tell you anything about what the movement's going to do, what kind of movement's going to happen. So this is just showing you the distribution on the left, the distribution of attenuation directions for planar reaching movements when the monkey was making a movement away from the body or towards the body, and they're almost identical, both in direction and speed on the bottom. In the human, we saw the same thing. If he applies a low force or a high force, the distributions remain invariant. So they're basically condition invariant signals that just indicate the movement initiation. The one thing we did find was that we said, is there any difference in any property of the behavior when we looked at these two directions of propagation? And the only thing we could find was that the reaction times are slightly shorter for rostral-caudal attenuation versus caudal to rostral. So this is for planar reaching movements, tongue protrusion, and isometric wrist extension. There is a significant decrease in reaction time for one direction of propagation. Why that's the case, we don't know. Okay, so I teamed up with Nicholas Brunel and Ludovica Romano several years ago. We started a project actually working with marmosets, which I won't talk about today. Although I have a postdoc, Jeff Walker, who's working on the data with the marmosets and is finding interesting propagating patterns as well in the marmoset sensory motor cortex. So Ludovica developed this model of motor cortex, just a leaky integrated fire neural model of motor cortex. Consider the spatial layout of the neurons in the network, and created connections with a probability distribution that fell off with distance. But one version of the model incorporated an anisotropy in the connectivity. So on the top right, you can see this anisotropy in probability of connections, where you see stronger connections along the diagonal and then on the diagonal from bottom left to top right, and then the probabilities drop off much faster along the orthogonal direction. And with this distribution of probabilities of connectivity, Ludovica wanted to look at what do we see in the local field potential in this model. And so in this case, what Ludovica is implying, and this is work done by Nicholas previously, is that the LFP is represented as the aggregate local AMPA and GABA currents onto excitatory pyramidal cells. And the LFP is also distance dependent, so it's not just, it falls off with distance. So it's basically the LFP, the local AMPA and GABA currents serve as a proxy for the local field. And with this model, incorporating that anisotropy and connectivity, they were able to recapitulate many of the findings we found in the motor cortex of macaques, where The recordings are shown in on the top, and simulations also show this propagating waves, phase waves during movement preparation. And they also propagate along one of two directions, as shown in the on the right. If you look at the autocorrelation functions, again, you see very similar patterns with oscillatory wings during the preparation of movement, just like you saw in the data. Now, what Ludovica then proceeded to do was inject input, external input to the model to denote sort of a go signal to the network, and that's shown in the top left. And sure enough, when she injected that noise, that input, and that input is actually homogeneous, but with added independent noise, you see a drop off in the beta power on a given electrode, and you see the autocorrelation functions during this period after the go cue, and that is the sort of simulated execution period when the monkey's actually moving. Very similar autocorrelation functions, and more importantly, she found that these attenuation envelopes propagate just like in the data. So in the simulations, you can see the blue squares attenuating earlier than the orange squares or the yellow squares, very much like we see in the recordings. So I think what's interesting about this model is that it incorporates a few things about the motor cortex, including this anisotropy, which has been shown anatomically and physiologically to exist, this sort of rostro-caudal anisotropy of connectivity. And from that, she can model the phase waves. And then riding on top of these phase waves is this beta attenuation as soon as you inject homogeneous input to the model. Okay, so that's all I have for initiation. Let's move on to execution. This is more recent work, and we wanted to find evidence of spatial temporal patterning that would actually carry movement information. So we looked at the high gamma frequency between 240 hertz, and we chose that because previous work, primarily in the BCI area, found that signal carries a lot of information for brain-computer interface work. And it serves as a useful proxy for multi-unit activity. And we showed that in this study. This was done by my former grad student, Liang. And she found that the high gamma is tuned to movement direction very much almost identically to what you see with the multi-unit activity. And we chose to use the local field potentials as opposed to unit activity because we could see it consistently across all electrodes, whereas we didn't always find multi-unit activity on every electrode. So with that, we can now have a good sampling of space across the array and then look for patterning. So if you look at the trial averaged high gamma envelopes, this is now from one electrode, and you can see that they carry information about movement direction. We had a monkey making reaching movements to one of 8 directions, and you can see how the envelopes vary as a function of movement direction. Wonderful. That's trial average, though. If you look at single trials, it's a big mess. So Wei had to figure out a way to clean those signals up, and she ended up doing a number of processing steps, including an autoencoder, contractive autoencoder, to clean up the signals. So this is the raw signal on the left, really a big mess. You denoised it with this autoencoder, then low pass filtered it, and then from that we can look at the we can now look at these now amplifications of the high gamma at movement execution and see what kind of information there is. Now, as I said previously, many people have found, particularly in BCI context, that the amplitude of this high gamma signal carries information about movement. But what we wanted to do was look to see whether the timing at which the amplification occurred formed a pattern, spatiotemporal pattern, or spatial pattern, and whether that carried information. So here's an example where a monkey is making a movement from the center target up and to the left to the gray target. And what we did here was look at the timing of the amplification, when the high gamma amplitude started amplifying across the array. And you can see here, maybe hard to see in 3 dimensions, but basically we could fit this with a plane, and from that we could then estimate a direction of propagation of the high gamma. The bottom example is for a movement down and to the left, and we see it likewise variations in the amplification time, and but with a plane and with a plane fit to it, but now oriented in a different direction. So we found that single trial propagation directions carried information about movement. Sorry, I don't know, this is going on by itself for some reason. Sorry. So we found information about movement in these propagating patterns. And if you look at many trials, for example, if you just look at movements to the brown target to the bottom left and the blue target to the top right, we can on the right you can see the direction of propagation across different trials presented on a polar plot, and the position of the dot along the radius indicates the strength of the fit of a plane. So so anything beyond this darkened black circle was significant based on our shuffling test. So we found, not in all trials, but many trials, significant planar fits to this propagating pattern. And moreover, this propagating pattern direction varied with movement direction. Now, what was interesting, we found something puzzling. So if you look here, if you look at the mean direction of propagation for the 8 different directions, at least for this monkey BX, we found that they clustered into 2 clusters. So a bunch of directions propagated to the down and to the left, and another set of movement directions propagated up to the right primarily. Now, why is that? It was kind of puzzling to us until we realized we had to overtrain the animal to perform movements either to the blue target or to the brown target. So what happened was, if you now look at what the monkey's actually doing from the center target, he started to move. He was overtrained to hit the blue target. He was also overtrained to hit the brown target. But the targets surrounding those 2 targets, what he tended to do is move towards the blue target and then shift direction. You can see that with the red one. So he started heading towards the blue and then he shifted. Same for the green. He started heading towards the blue target and then he shifted to the right. So the kinematics sort of naturally segregated into these 2 clusters, and this to us suggested that if these propagating patterns carry any information, they may be carrying information about the initial launch direction and not continuous movement throughout the behavior. So it's just that initial launch direction. So we built a decoder, a simple linear decoder, and we could, that is, decode from three parameters. So we did not consider the amplitude of the high gamma. What we considered was the propagation direction, how well they were fit to a plane, and the propagation speed. And with those three parameters, we could predict the velocity of the hand with up to an R squared 0.4, ignoring the amplitudes. So, which was kind of impressive to us. And most of that information is carried by the propagation direction. So again, we're ignoring amplitudes, just looking at the propagation direction. And then finally, we wanted to ask the question of whether these propagating patterns carry redundant information with the amplitudes or carry maybe additional information. So if you look at just propagation, as I said, we can predict the velocity of the hand with an R squared of about 0.4, a little higher. That's shown in the red horizontal line. Now, if you look at the amplitudes of the high gamma only, it shoots way up to 0.7 or higher, which again, previous people have shown. Now, if you include both the high gamma amplitudes and the propagation parameters, you get a slight boost. It's significant. It maybe not that dramatic, but still you get a boost in performance in the decoder. So you can actually predict more accurately the velocity of the hand. So that's pretty much all I've got. I think where we're heading with this is, well, actually, I want to thank my group here. Where we're heading here is to develop this model further that Ludovica and Nicola are developing to make testable predictions that we can go back into and do experiments to test some of these predictions. We also want to understand exactly if these propagating patterns during execution are real, which they seem to be. How do they emerge in the cortex? Well, I mean, why do they emerge the way they do? And one idea we have is that when you make different movements, you might be recruiting muscles with different sequential activation of different muscles. So, for example, if I go back, so if you take, for example, the monkey's starting from the black center target and he's going to the blue target, you see this propagation down and to the left across on the cortical sheet. Would that also would that imply that muscles are being recruited sequentially? Muscle representations in cortex being activated sequentially, depending on the movement that the monkey's making. So the way to test that would be to stimulate each electrode, assess what muscles are being activated with stimulation on the cortical sheet, and then see if, in fact, that when let's say the pattern is propagating down and to the left, in fact, the cells are activating in a way consistent with the sequence of muscle activations. So that's where we're heading right now, and I think that's all I've got. Thank my group, and take some questions.
Thanks so much, Nicho.
Thank you.
If you guys have questions, you can post them in the Q&A feature. We're having a couple of technical problems today, so I hope that is working for everybody out there.
I have one question meanwhile.
Yeah, go ahead.
Wonderful talk.
Thank you.
The gamma stuff are really exciting. I was wondering, what do you think about the nature of these waves that you measure, following up to the beta waves in the neuron paper? Do you think these are constant type of oscillatory waves or more of a stationary type of activity? I remember the beta paper was more of a short transient type of beta, and I was wondering if the gamma one is also similar to that.
Right. Yes. So with the gamma, well, you're talking about, yeah, so you're talking about the amplitude of the
Yep.
of the gamma, right? So yeah, I think it is transient. And in fact, we're testing this idea with a more complex behavior. So the monkey now, instead of just making a reaching movement to one target, he's making reaching movements to a whole bunch of targets. So he's basically kind of scribbling. And what I mean, this is a working hypothesis, it may be wrong, but what we might see is sort of these transient increases in high gamma that fall down and then come back up. Let's say when he moves, makes a correction or makes a new movement to a new target, and so you basically have almost like an intermittent signal that's controlling. I mean, this is a wacky idea. I don't know if it's right, but that maybe the core. And if there were any motor physiologists in this audience, they'd probably laugh at me. But anyway, this idea that the motor cortex might be providing intermittent signals, and actually not providing continuous control. And much of the continuous control is happening subcortically. Now, of course, there are continuous signals in the motor cortex. I mean, we see that, and there's information there continuously. But maybe that information is sensory in nature, because the motor cortex also receives sensory information about the ongoing movement. But the commands might be just intermittent. And so basically it's almost like a ballistic movement. You create this transient command that's signaled by this transient high gamma pattern, not just the pattern, but also the amplitude. And then that moves the arm in a certain direction until there's either a correction that it needs to be made. Yeah, some correction needs to be made, and at which point a new command comes on and you see another increase in high gamma.
I see.
Nicho, is the thinking there that like each wave is like a packet of information, a command? Is it that?
Yeah.
That's
Yeah,
how you're thinking of it?
exactly. And it's intermittent, which is, yeah, it's not widely accepted, and I'm not sure I believe it, but it's so.
That's very honest.
But that's what we're working with, the idea, because there's work done, Yifat Prut in Israel. I just remember an old study where she recorded simultaneously from the motor cortex and the spinal cord interneurons, and for a reaching movement, just the center out reaching movement, and she showed that the motor cortex tends to be more transient in its activation. Actually, I think this was an isometric force task. Anyway, so there's very transient activity in the motor cortex. The interneurons were more sustained. So suggesting that maybe that the cortex is providing this sort of intermittent signal to sort of trigger the interneurons and ultimately the motor neurons to activate. And then the spinal cord sort of maintains that until a correction needs to be made.
All right, I'm gonna pull someone up from the audience for the next question.
Hello.
Good, whenever you're ready.
Okay. Thank you for this wonderful talk. And I have one question regarding the movement execution study using the high gamma power. So I wanted to know, what parameters did you use to detect the amplification of activation?
What parameters did we use to measure the timing?
Yes, like was there any threshold or like
Right. So we started with a threshold, but we found that to be kind of arbitrary. So then what we did was we took the first derivative of the envelope as sort of a proxy of like when this and looked at the peak of the first derivative. So that's how we ultimately determine the timing of that attenuation. Like when it came on was when the first derivative was a maximum.
Okay. And that you considered for all the electrodes, right?
For all the electrodes, yes.
Okay. Thank you.
Can I ask a follow-up question to that?
Sure.
Israt, thank you for your question, by the way. And I had a related question, which is about you mentioned the autoencoder, and autoencoders are powerful things that can do a lot. What exactly was it doing in this case, and why wouldn't it have been? Why there must be a reason you had to resort to such a powerful method, right?
Yeah. Well, I mean, it's
Like what was it doing? Yeah.
basically what it's doing is it's compressing. It's like a bottleneck. So it's compressing the data, the high dimensional data. So basically we feed it all the channels and all the times, and then feed it through this hidden layer that's basically contractive. It's compressed to sort of remove the noise.
But I thought you said that there was one number coming out of there, or is it a multi-dimensional thing? Was it like a lower dimensional space, but not all 100 dimensions? Or...
No, it's an autoencoder, so what comes back out is the same signal.
Oh, just rearranged.
But then but cleaned up, essentially. It's like
is it like a low pass? Are you using it in this context like a low pass filter, like an averaging?
Yeah, it's like a low pass filter, but if you just apply low pass filter to the data, it failed.
Okay.
And so it's like using PCA. You use PCA, you then keep the first few principal components where you think the remaining is kind of noise, and then use those. Or it's sort of like that.
Okay.
Yeah.
Great, thank you, Israt. Bard had a question as well.
Yeah. So I'm curious if, okay, you train the monkey only on those two directions, but then when you had him do other reaching, it moved, it clustered into only those two directions. And I guess that's because of the intrinsic anisotropy and the motor cortex. I was just curious if you didn't like overtrain it on those two directions, but trained it equally on all directions, whether you'd get more variety in the traveling waves. And also, I'm trying to understand what the utility of these waves are in terms of the sequential movement.
Okay.
And why the sequence, if I'm reaching up here, would be opposite the sequence if I'm reaching down there.
Right. Yeah, so the first question is that the anisotropy and connectivity that Ludovica and Nicola put into their model has nothing to do with the training of the animal. I mean, that's based on previous work.
No, I know. I mean, that's why my curiosity was
Yeah.
whether you only get waves along that axis,
No.
if you trained equally, would you see wave in other directions?
Yes, that in fact in other animals we did see movement propagating directions in other directions. So it's not just unique to those two directions, right?
And is there any correlation with those directions with the direction of the movement, or is there any relationship of the direction of the waves with the motion direction of movement?
Well, that's on some level, yes, because we found that we could decode the velocity of the hand with.
That's right. Yeah, that's right.
So we could decode. So there is some correlation. Absolutely. The other question, which is tougher to answer, I don't have a good answer for that. Why would a movement, let's say, if you go back to the, let's see, why would a movement to the blue target result in a completely opposite direction of propagation versus the brown target if it's muscle recruitment? Well, if you think about what the animal is doing, the blue target is away from the monkey's body. So it's involving, it's a lot of extending the arm out from the body. The brown target is moving towards the body. So it's a different, you're activating different flexors and extensors and different patterns.
Oh, maybe it would help if I knew what the orientation of this was with respect to the monkey's body.
Oh, okay. Yeah. So
Monkey looking down at it, and it's
yeah,
like a
right.
Ouija board or something, or a bull's.
Well,
Or is
it's
it-
actually okay. Okay, so there's this. You see the screen with the dots, right? The
Yep.
the dots are presented actually on a vertical screen in this experiment. Okay, and so he's sitting in front of this screen, and to make a movement, like to the red target, he basically has to move his arm forward away from his body.
Okay.
And
I
the blue target is he moves his arm forward and a little bit to the right,
see. And the orange one, he's actually moving it directly to himself.
directly to himself.
I see. So he has like a little lever, or he's holding on to a joystick or
Yeah,
something?
he's holding onto a joystick.
Okay. Okay. Sorry, I didn't know how these experiments worked. Okay.
Yeah. So right, I don't really have a good answer for you, Bart, but yeah, these are different kinds of behaviors. One, I'm moving away from your body.
Yeah. Okay.
Extension of the arm muscles and versus moving towards your body.
All right. That makes sense.
Yeah.
In a sense, a little bit. Yeah.
A little bit. Yeah. And people have looked in humans making reaching movements like the monkeys did in the plane. And this is a study by Hassan where they looked at they recorded muscle activity and looked at the temporal order of activity. I have to go back and look at those studies and see exactly. But the temporal order of activating muscles varied with the direction of movement. So you get different sequences of recruitment of different muscles depending on the movement direction.
Just one more quick, totally wacko question. Okay. If you imagine like a seizure, like Jacksonian seizures that propagate across the cortex.
Yes.
Okay, I guess that activates, you'd see the same motion in the muscles, in that Jacksonian march and things like that.
Yeah, absolutely. Yeah. In fact, that's how it was first documented, as far as I remember, as far as I understand. That's
how they first discovered those somatotop. I mean,
they discovered it was somatotopic.
Yeah.
Okay, great. I'll pull up one more person from the audience.
Hey, thanks so much. This has been an excellent talk. I was wondering if you looked at the beta band during the motor execution studies, and vice versa with the high gamma during the planning studies.
Yes. Well, we definitely looked at the beta during the execution, and basically we found no. Well, first thing you should know is that even though the beta amplitude attenuates during execution, it's still there. You actually do see waves even during execution. They're just highly attenuated. But we did look at, I remember, we definitely looked at information about movement with the beta. And I'm trying to remember what I think we just looked at the beta amplitude, and there was like no information. There's no information about movement in the beta frequency, whereas there is quite a bit in the amplitude of the high gamma. Now, during preparation, yeah, we did not look at high gamma during preparation. Again, what we see is this dramatic, we see this dramatic amplification of the high gamma at movement when the execution begins. But there's still probably, there's still high gamma signal there during preparation, but we didn't look at it.
And then a follow up to the overtraining the animal hypothesis. If you looked at earlier trials versus later trials, as the animal learned the tasks, did that kind of prove your hypothesis correct, where you saw multiple directions, or you were less likely to see two clearly distinct directions?
Right. That's a super cool question. And no, we didn't look at that. I mean, part of the problem is that, I mean, we need a lot of trials, and when they're first learning, we don't get a lot of trials out of them, but that's a super interesting question. Which, during the early phase versus the late phase. It's not that they were not trained on the other movements, but they were more often on certain days. We said, let's just focus on those two targets and ignore the other ones. On other days, they had the other targets as well. Because we were trying to up the number of trials for each target direction. So we said, let's just reduce the number of targets to just 2. So
that makes sense.
Anyway, but that's a super interesting question during the learning process to explore. I haven't looked at that.
Cool. Thank you.
Sure.
Any other questions?
I kind of have a follow-up along the lines of the overtraining hypothesis. So you had that diagram where it starts, specifically in blue and brown, and then branches out.
Yeah.
Later in the movement execution, do you see something different? Do you see like not specifically
Let me just get
waves
that.
along those two axes?
Right, hold on a second. Let me just right here. Yeah. So what was your question?
I guess later in the execution, do you see more like diverse propagation directions that aren't specifically along those two axes? Like maybe they correct or
Right.
they,
Yes. Yes. Right. So we did do that. So for example, my current postdoc Milan now is looking at, do you see the screen?
Yeah.
Yeah. So for example, look at the red target.
yeah.
So he's moving in one direction and then he corrects.
Mm-hmm.
And he shifts to the left. So he's looking at that in time. But
Yeah,
he's not, but now he can't look at, because he's looking at every moment in time. Instead, what he's looking at is just space. At every moment in time, what is the is there a spatial pattern of amplitude?
Okay.
You know what I mean, so every moment in time, you said, okay, what's the amplitude across the array? Does it form a pattern?
Yeah.
And in fact, he found, interestingly enough, support for this intermittent hypothesis, where there's spatial information at the beginning of movement, and then it gets weaker and weaker. And then at the correction, it increases. That is, there's spatial information during the corrective movement, at the beginning of the corrective movement. I wish I should have shown you that, but anyway, yeah.
That's really cool, like almost like a recalibration.
Yeah, exactly.
So there are transient moments when there's information about in the spatial pattern of the amplitude,
Spatial
but only
pattern-
at certain moments.
only at certain moments, exactly.
It's like the Walter Freeman kind of patterns, like those amplitude patterns, if you're familiar with his stuff, or
I don't remember exactly.
yeah, I mean, it sounds like it's exactly those things, the patterns he talked about
Yeah.
in a different setting, but
Yeah.
but it's transient. Yeah,
It's transient. So that's actually what led us to this sort of intermittent hypothesis, was the spatial patterns that we saw that came on at certain moments in time, and they carried the spatial patterns, carried information about the movement at the beginning of movement, then they shut off, they get much weaker, they can't predict, then they come back on at that corrective phase.
is there any other way to predict when that information is present, like from some neural signal? Like, let's say that we didn't have, like, the reason I'm asking is because of some of our human I'm interested in looking for these patterns in humans.
Mhm.
That's what we do in my lab, but we don't always have quite as well characterized behavior on a time scale of milliseconds as in
Yes.
movement. So I'm wondering if there's like another internal brain signal that one could look at to decode those things,
Internal brain signal as opposed.
like coupling to some slower oscillations, and that's when the patterns show up or something like that.
Oh, I see. Yeah, I don't know. I can't think of anything.
Okay.
But yeah. Some coupling to another off frequency or something,
Or some other marker, yeah. Okay.
some other marker. I mean, there might be something in the, there's some work done by Mark Churchland suggesting that, with a multi-step movement, you make a move, you plan, you make the movement, and then you switch to sort of a preparatory subspace, which is almost orthogonal for the next movement. So you can see that switch when he's like, okay, I made the movement to the first, now I'm going to switch to prepare for the second one. And
that's the moment when you should see it. Yeah.
Yeah. So you could see that in the population activity by looking at like null spaces, activity null space during that. Yeah. Yeah.
Cool.
good.
Uma and Irfan, should we wrap up? Is there anything else? I can't see the question list because of these issues I'm having technically today, so that's why I'm checking in.
Yeah, I think that was it for questions. Thank you guys so much.
Thank you.
Nicho, this is wonderful. Thank you so much.
Yeah. Appreciate it. Yeah.
Great to see you, Nicho.
Good to see you too, Bard. I'm glad you made it.